Hey everyone, welcome back. In today's video, we're going to talk about SAM and BAM files. These files come up a lot in bioinformatics, so definitely something you're going to want to know about. Let's get started. So what is a SAM and BAM file? It's a file format for storing sequence alignment information. Now both SAM and BAM files store the exact same information, but SAM files store alignment information in a plain text format, while BAM files store alignment information in a compressed binary format. And in practice, you'll mostly be working with BAM files because they take up less space than a SAM file. And you'll know when you see a SAM file because they always end with a .sam file extension, and BAM files with a .bam file extension. Now, where do SAM and BAM files come from? They come from taking the sequence reads in our FASTQ files and aligning them to a FASTA formatted reference genome. And returning to our puzzle analogy, it's sort of like taking pieces to a puzzle and comparing them to a completed version of that puzzle. And information about where the pieces go, how well the pieces fit, and which pieces don't fit at all are stored in the SAM and BAM files. And this entire process is referred to as sequence alignment. And similar to genome assembly, there's an entire field of bioinformatics dedicated to just this topic. And I'll make sure to provide some links to useful papers about sequence alignment in the video description for those interested in learning more about it. Now, what is the SAM format? It's a tab-delimited format consisting of 11 mandatory fields. And these fields provide us with information about both mapped and unmapped reads. When I say mapped, I'm referring to reads that aligned somewhere in the reference genome, and unmapped meaning the reads didn't align at all to the reference genome. There's also optional fields which contain extra information about each alignment, such as whether or not the read mapped to multiple locations in the reference genome, or the presence and location of base modifications. In this video, we're just going to focus on the mandatory fields of the alignment section. And this is a table from the official SAM file format specifications, and it describes each of the 11 mandatory fields in detail. Now this table might look a little scary, but I promise you it's not that bad. Let's look at a small SAM file and go over each field individually. This is what a SAM file might look like if you were to view the first few lines of it. Each line corresponds to an individual record within the SAM file. In the first column, we have the name of each read that was processed, and each read name will be identical to those found in the input FASTQ file. The second column is an integer, which encodes specific information about the alignment, such as whether the read mapped to the reference genome at all. The third column contains the name of the reference sequence that the read aligned to. This could be the name of a chromosome, gene, bacteria, and so on. The fourth column is an integer specifying the starting position of the alignment with respect to the reference sequence. The fifth column is an integer describing the mapping quality. Higher numbers indicate better mapping quality than lower numbers. For example, a read aligning uniquely to the reference genome might have a higher mapping quality than a read aligning to multiple places in the reference genome. The sixth column is called a cigar string, and it provides information about the structure of the alignment, how many matches and mismatches, insertions, deletions, and so on. The SAM file also contains information about the mate pair, which is applicable in paired end sequencing when you're sequencing two ends of the same molecule. And the seventh column tells us which reference sequence the mate pair, or the other read, aligned to, and the eighth column tells us the starting position of that alignment. The ninth column is an integer that represents the length of the fragment of DNA or RNA that was sequenced, and we'd expect this to be roughly the same across each library, depending on how the genetic material was sheared. And the 10th column is simply the nucleotide sequence of the read that was processed, and the 11th column contains the quality scores assigned to each nucleotide within that sequence. And I know this is a lot of information, but I think it's good to have some idea about what's present in these files, because it gives us some intuition about what types of analyses we can do with them. And for those of you who are interested in taking a deeper dive into the SAM file format, I would definitely recommend checking out the official SAM format specification. I'll provide a link in the video description. It has a lot of great information and will be especially important for those of you who might be writing programs or scripts to parse the records in a SAM and BAM file. Now, what are SAM and BAM files used for? They're used for a few different things. One of the most common use cases of SAM and BAM files is to store both sequence and alignment data. You may have noticed that the SAM and BAM files contain everything a FASTQ file does: the read name, nucleotide sequence, and quality scores. This means that FASTQ files can be recreated from just the SAM and BAM files. So what you'll often find in practice is that once the bioinformatic and data analysis is complete, you might discard the FASTQ files, but archive the SAM and BAM files on a remote server or with a commercial cloud provider as shown in this example. The second use case of BAM files is for identifying sequence variation. BAM files are often used as input into variant calling programs like GATK, Freebase, and somatic variant callers like Mutect2 for identifying single base substitutions, insertions, deletions, and other complex variants. Third is quantifying gene expression. BAM files are often used as input into tools like feature counts or HT-seq to count and compare the expression of genes between experimental groups. And fourth is for visualizing genomic regions of interest. BAM files can be used as input into genome browsers like IGV, which can give you an idea about how many reads did or did not align to a specific area of the genome, and many other use cases. So where can we download SAM and BAM files? If you're reanalyzing data, it's definitely more convenient to begin the analysis with the BAM file, because it contains all of the raw sequence data and alignment information, which you won't need to regenerate again. And there are numerous online databases where you can find these files. Perhaps my favorite is the Genomic Data Commons, because they provide access to high quality and curated cancer science datasets from numerous different projects. They also have standardized bioinformatic pipelines that run through every dataset that is submitted to their database, and they have excellent documentation. I would highly recommend checking them out if you're interested in cancer science. There's also the Gene Expression Omnibus, GEO, the Sequence Read Archive, SRA, and the Registry of Open Data on Amazon Web Services, AWS, which is becoming a popular place to park sequence data, especially for large-scale projects. AWS will often store this data for free on the condition that it's made publicly available, which is nice because the download speeds from AWS are quite good compared to other sequence databases. Now let's look at a real-world example involving a cancer called multiple myeloma. Multiple myeloma is a cancer of plasma cells, which are found in the bone marrow and play an important role in the functioning of our immune system. And some of the questions that researchers are still interested in learning more about is why some people respond to treatment while others don't, and if there are unknown genetic markers that play a role in the progression of multiple myeloma. And to help answer these questions, the Multiple Myeloma Research Foundation funded a study called CoMMpass, which was a longitudinal study that enrolled 1,154 individuals with multiple myeloma. And researchers collected bone marrow samples at baseline, during treatment, and periodically during an 8-year follow-up period, and the DNA and RNA from those bone marrow samples sequenced. The FASTQ files were then aligned to the human reference genome, which produced a SAM file, which was converted to a BAM file. And this BAM file was used for two types of analyses. The first, gene expression quantification to identify multiple myeloma subtypes, and second, to identify sequence variants responsible for loss of function or gain of function gene mutations. And these results are helping scientists learn more about the biology of multiple myeloma. And if you'd like to learn more about this study, please check out the open access article in Nature. I'll post a link in the video description. That's it for this video. In the next video, we'll be talking about an alternative file format for storing sequence alignment information called CRAM format. You won't want to miss it. I'll see you there.